if you say the globally, that where it's very difficult to degrade the plastic. So he will be highlighting some of his research points where his lab is working on the role of biofilms in the plastic degradation. So with these words, I invite Professor Antaresh Kumar to deliver his talk.
So, good morning. Today we have selected a new topic, that is the role of biofilm in the plastic degradation. So far you have discussed different aspects of biofilm in pathogenesis, in other aspects like food processing. So today's topic is related to that role of the microbial biofilm to degrade the plastic. You all know that plastic is the major problem worldwide, which causing a different kind of pollutions in our ecosystems. So key questions came into the mind that when we deals about the plastic, how big the plastic for problem is, how much plastic is we generally generate, and how much plastic we consume, and what are the management of the plastic, whether the plastic which we produce is actually recycled, or how do we manage the plastic after this. Second question is how much plastic we recycled, the plastic which we generate, that what amount of plastic we recycled and reuse it again. And the third and most important point is how do we manage the plastic pollutions or plastic waste which we generate. So the today's topic is related to the third questions which we have in the mind, then how do we manage the plastic waste. So if we talk about the plastics, overall the worldwide plastic we produce around 415 million metric tons generally plastic generates all over the world. And out of this 415 plastic, 360 million metric ton plastic we generally trash into this, generally trash it, which is approximate 87% of the total amount we generate. So this is a major problem in the plastic, we use it and throw it into the trash. And the third one is how much amount of plastic we recycled worldwide. So approximate 17% of the total amount, which is equivalent to around 62 million metric ton we recycled metric ton, which is 23% of the plastic we doesn't care about it after use it. So that causes the problem in our ecosystem. Problem major in the mismanagement of the plastic, we use it and throw it here and there. That causes the problem and causes the pollution in the environment. So these are the different countries worldwide produce or consume the plastic, and we stands at the third position after China and US. And the contribution in our terms of the global, that is 6% we generally produce the plastic all over the plastic generates around the globe. Plastic after the use we generally throw in the different areas, particularly the primary site where we can throw the plastic is lands. And if you look at the data, approximate 50% of the plastic which we generated generally fell into the lands, that is approximate 178 million metric ton plastic, generally landfill plastic, which creates a waste or pollution, so generates the gases after degradation and disturb our ecosystem. Along with this landfill or soil, we also throw the plastic into the aquatic systems. And if it is look at this UNESCO data, approximate 100 million metric ton plastic is already present in the ocean, and per year we generally throw this 17 million metric ton plastic into the oceans, which disturb flora or fauna of the aquatic systems. how this aquatic system get disturbed by the plastic waste. So the plastic which we throw into the aquatic system gets converted into the microplastic by different means like biofilm, microbial biofilm, some sewage treatments and some UV degradations. And ultimately this plastic converts into the microplastic, will be ingested by either the some of the fishes or some of the aquatic organism, and also adhere to the algae or some plants present in the area in the aquatic system. And this microplastic ultimately goes into the food chain, in the human food chain, by different ways. Not only in our case, it also goes to the some of the the birds which we can generally attack or eat or take this fish as a food. So in that way, the plastic which we throw into the aquatic system ultimately goes to the human as well as also goes to the different species and cause the pollution, and it shows the detrimental effect onto the human metabolic system as well as their bird metabolic systems. So these are the different worldwide studies. There is a study that they have tested the different freshwater rivers, the plastic content present in that rivers. So they have checked in the China has this, they have taken the sample of this particular reservoir, and they have found there different type of plastic like polystyrene, polypropylene, polyethylene in different amounts. Even though in the Vietnam, Portuguese, Brazil, and look at this is the India, northwest India, particularly the Ganga or Yamuna is also get polluted with the plastic, having a different type of plastic presents that affect the overall flora and fauna of the water system in India as well. What are the impact of the plastic if it contaminated our land, aquatic system or air? The plastic, if it is present in the soil, can affect the soil fertility, can deteriorate the soil quality, and as well as if it is the plastic is present in the soil, after this the degradation, it produces some kind of gases which is toxic to the environment and participate in the global warming. In addition to that, the plastic underleach into the aquatic system, that may also affect both the productivity and the growth of the aquatic organism as well as the flora and fauna of the aquatic system. So overall, the plastic, if it is present in the soil system and the plastic waste present in the air as well as in the aquatic system, will produce a detrimental effect all over the ecosystem. So if we talk about the plastic, majorly are two type of plastic, as you know. One is the thermosetting plastic, another one is the thermoplastic. Thermosetting plastics are those which can heat it once and then it converted into the hard structure, hard substances which cannot be remolded to if we reheat it again. So such kind of plastic is called as thermosetting plastic, and these are the different example of thermosetting plastics. That urea formaldehyde is used as a thermosetting plastic, which are used in the electrical fittings, makes and knobs and some additives. Polyurethane, examples of automobile interior, make a foam insulator layers, is a polyurethane. Phenol formaldehyde, bakelite, that makes a electrical switching, machine parts of the electrical systems. And also epoxy resins, which also used in the casting and encapsulation adhesive and also other materials. Bakelite is also used for the electric switches and other electrical versus cookwares. So these are the different examples of the thermosetting plastics. And next category of the plastic we can is that it's thermoplastic. Thermoplastic is the main one which we generally use in our daily life, like polyethylene, polystyrene or some other type of plastic we generally use in the daily life. So these are the example of the thermoplastic, like polyvinyl chloride, we generally use the pipes we made up of this PVC, credit cards, tiles and also pipes. Low density polyethylene, that is LDP, that also used in the garbage cans, frozen foods and plastic wraps is also made up of this low density polyethylene. High density polyethylene is basically the milk bottles, bleach type bottles, sparks, and other articles are made of this high density plastics. Polypropylene, that Tupperware, car parts, yogurt containers are made up of the polypropylenes. Polystyrene, the egg cartoons, disposable dinnerwares. Nowadays we generally use in the marriages the plates which are basically made up of the polystyrene will also produce the pollution after use it and throw it into the soil. Polyethylene terephthalate, which forms the food tray and beverage bottles, these all are the example of the thermoplastic which contributes majorly into the plastic waste in our daily lives. So, if we look at the Indian scenario of the plastic we generally use, so overall the India produce the total 25 million metric ton plastic per year, and the total waste we generate around 6.6 million metric ton, which is almost the 26% of the total amount we generally produce of the different type of plastic. And the mismanagement of the plastic in India is the very worst condition. The plastic which we use it generally throw it, and in that case India is the top position to mismanage the plastic. After use it can throw here and there, and very less amount of plastic gets recycled. Around 3 to 5% plastic we recycled and reuse it again after the once time. And the most striking data is Delhi is the highest generator of the plastic waste. It is the only city all around the India which produce the highest number of the plastic waste, that is around 1100 metric ton per day. That much plastic is produced by Delhi itself. So to think about how to manage this plastic, so we generally consume India level, there are different type of plastic we consume in the different ratio. For example, we use in India polypropylene we use approximate 33%, polyethylene we use approximate 30%, and 70% we use the PVC for the pipe and other purposes in India. And rest that contributes around 18 to 20% in the total of the plastic we use. The major problem in the plastic is the conversion, because the conversion or the decomposition of plastic is very hard. It takes time, and in the composition that the plastic which is the convert first into the microplastic or nanoplastic produce the major pollution that ingest by the organism like plant or aquatic system. So the recycling and conversion of the plastic can be done will be three different means. It can be done either by the chemical method can be used to convert the plastic into their last product like monomers, or can be used that monomer to recycling purposes. Another one, we can use the biological approach. The biological approach can be used, some plants can use a microorganism, fungi that can convert this macromolecule or polymolecules into the simpler form. And third one is the hybrid mode. We can use some chemical or environmental or thermal condition or physical condition along with the biological condition can upscale the plastic recycling. The upscaling of plastic recycling is related to we can reuse this plastic some other purposes, and I mean the plastic can be used for the second time or multiple time by upcycling. So as compared to the chemical method, I am not going to discuss in detail. The pyrolysis is the one way we can convert the plastic into the gaseous form. Depolymerization and gasification is another method of the chemical methods. We are more focusing in this biological method of the plastic utilization or recycling or decomposition or conversion of plastic into the simpler molecules. So if you talk about the biological method, first microorganism came into the mind because that is the natural process that microorganism tends to adhere onto the surface of the plastic and they change the plastic. Plastic is basically a hydrophobic in nature. So for microorganism, either bacteria or fungi or some other organism, they tend to adhere or interact with the plastic will first try to convert the hydrophobicity, reduce the hydrophobicity by changing some functional group present in the plastic. So microplastic, basically a microorganism biofilm first adhere to the plastic and convert the plastic into their less hydrophobic structure. So once the microorganism adhere to the plastic surface, it tends to release some enzymes, it tends to make some extra polysaccharide polymeric substances which can change the functional group present in the plastic, and in that way it can change the reduce the hydrophobicity of the plastic. So microorganism biofilm play a critical role for the adherence. Not only adherence, but it also has a role into its degradation by stepwise changing the hydrophobicity of the plastic into the hydrophilic nature. Once the hydrophilic is nature, then it gets digested or decomposed by the microbial enzyme. So this is the overall mechanism of the plastic, that microorganism biofilm tends to help in the adherence to the plastic. Once the microorganism biofilm adhere to the plastic surface, it tends to change its hydrophobicity, reduce the hydrophobicity by converting the polymorphism functional group into the hydrophilic groups, and these hydrophilicity increases that helps to facilitate the acting the enzymes present in the biofilm region to degrade the plastic into the simpler molecules. So microorganism quickly form the plastic, and the microorganism grown in the plastic term is called as the plastospheres. So the plastosphere microorganism forms the plastospheres that acts as a miniature ecosystem, that acts as the small ecosystem that helps to degrade the plastic by releasing a different type of enzymes. So how this biofilm formation occurs onto the plastic by different microorganisms. So there are different steps are involved in the biofilm formation. First step is the adherence or attachment of the microorganism to the plastic. So this is a microplastics, and these are microorganisms that they adhere, and initially there is a certain molecules expressed by the microorganism, like biofilm is active, certain EPS molecules is also synthesized that helps, that facilitate the microorganism to the adhere onto the hydrophobic plastic surfaces. So first step is to the attachment that provide the hydrophobic nature of the biofilm of the microorganism, facilitate to adhere or bind or attach to the plastic surfaces. The binding of plastic surfaces depending upon the nature of plastic, whether it is the polyethylene, polystyrene or some others. Another nature is how this EPS is releases, amount of EPS, amount of enzyme is releases that provide the interaction between the plastic to the microorganism. So after the microorganism gets attached to the surface, the next step of the biofilm formation is biodeteriorations. As the name suggests, there are a certain number of enzymes is releases after the attachment of the micro to the plastic surface. These enzymes tends to secreted extracellularly that can adhere, that converts the plastic into the different monomers or different forms. Particularly in that particular stage tends to start the hydrophobicity alteration. I mean the hydrophobicity tends to reduce by attaching, by releasing some enzymes at this particular stage at the biodeterioration steps. After the second step, the third step is this biofragmentation. As name suggests, the polymers of the bioplastic gets biofragmented with the help of the different type of enzymes releases, and the long polymer chain of the plastic can be converted into the simpler form by this particular steps, biofragmentation, and these converts into the simpler molecules of the polymers, like polyethylene converts to the ethylene or some other related compounds. These similar monomers of the plastic can be utilized by the microorganism for their growth and act use as a carbon source for their growth. So the next step, fourth step of the biofilm formation is assimilation or mineralization. This simpler molecule synthesized by the degradation or biodeterioration of the plastic. This simple molecule can be assimilated by the microorganism that can be used for their growth by different like abiotic or anaerobic or fermentation process of the microorganism required for the growth for the microorganism. Some of the molecules which are not utilized by the microorganism that also be non-assimilated foods we also synthesize inside in the culture. So both monomers which is assimilated by the microorganism or utilized by the microorganism or not utilized by the microorganism will also synthesize during this step. And ultimately this monomer convert to the carbon dioxide or water molecules as a mineralization form. So ultimately the plastic microorganism adhere to the plastic ultimately convert the plastic into the mineralization form like carbon dioxide in the water by getting metabolized by the microorganism cellular mechanism or may also convert some other molecules depending upon the type of plastic. There are different variety of the microorganisms have been reported that can be used for degrade the biofilm, degrade the plastics. These are the different genus. I am not going to discuss one by one. So these are the list of the genus or these are the different families of the bacteria will tends to degrade different kind of plastic like LDP, polyethylene, polypropylene, polyvinyl alcohol, polyethylene and polypropylene, polystyrene, etc. So there are a number of reports have been published related to identify the microorganism which can adhere to the plastic and degrade it into the simpler form. What are the factors responsible for the plastic degradation? So depending upon the biofilm, there are two types of major factors involved in the degradation of bioplastic by the microorganism. One factor is rate and quality of the biofilm formation, how much amount of PPS is formed, how efficiently cells grow onto the plastics. So all these factors affect the plastic degradation. And second one is the type or property of the microplastic, whether it is the polyurethane, polyethylene or PVC. If it is PVC, it is very hard to degrade as compared to P or polypropylene. So these are the two major factors. One is the nature of the plastic, what is the plastic, what kind of plastic it is, and second one is how what amount of the EPS or how fast the bacteria or the microorganism grow into the plastic will affect the plastic degradation. In addition to that, certain physiological factors or certain other factor will also affect the plastic degradation, such as the temperature. If it is little bit increased, will increase the metabolic activity of the cell, also helps to faster degradation of the plastic. pH is also one factor can affect the plastic degradation, microbial plastic degradation. UV is the another one, and the nutrient condition, limiting nutrient condition may also augment to plastic degradation by the microorganism. So in addition to those two factors, some other environmental factors will also help to degrade the plastic by different type of microorganisms. Let's take the one example of this degradation of polypropylene or polyethylene plastic. So before discuss into the detail, first we'll go through the outline and then go to the mechanism how it works. So this is, let's say, the polyethylene or polypropylene particles where the microorganism is grown attached there, and this is the chain of the polypropylene or polyethylene. So this chain can be altered with this hydrophobic chain converted little bit hydrophilic form. Hydroxyl group is hydroxyl functional group is attached to this. In that way, the hydrophobicity of the plastic gets altered by attaching the microorganism using certain kind of enzymes. So before discussing how this plastic, basically polyethylene or polypropylene, gets degraded, first we need to understand this polyethylene, is the polymer of ethylene, and there are different type of polyethylenes are present depending upon their branching chain. So the least branching chain is the high density polyethylene, which has the very short side chain, few or no branching. Another one is low density polyethylene which has the significant number of the branching as compared to the carbon-carbon chain. So, in addition to the carbon-carbon chains, the low density polyethylene has the higher number of branching carbon chain. So, this higher the number of branching, higher the flexibility and the lower the density. This is the relation between the flexibility. So, these are the this less flexible and the low density polyethylene is the more flexible. So, another one is the propylene is the polymer of propylene, this monomer of propane. So, there is a polymerization of this particular catalyst can convert into 23 to 35 carbon chain longs. It may be the arrangement of this propene monomer into this range, I mean has the regular methylene group attached. That kind of polypropylene is called as entactic propylene. Maybe the one sided of the methyl group attached there in the polypropylene. Such kind of propylene is called as isotactic polypropylene, which has the one sided methyl group, and some may have the propylene layer, the arrangement of methyl group into the alternative form. That kind of propylene is called as syndiotic polypropylene. So, if we know, once we know the structure of the polyethylene and polypropylene, so how does the polypropylene and polystyrene gets degraded? So, I told then once the microorganism adhere to the polypropylene or polyethylene surfaces, it first tends to increase the hydrophilicity. So, this is the polyethylene and the microorganism adhere to this, then it's form the formation of hydrolyzable groups. So, it converts the hydrophobic carbon-carbon in attached to the carbon-carbon backbone of the polypropylene or polyethylene to convert into the hydroxyl group attached by the action of certain kind of enzymes present in the biofilm area of the microorganism, and some can be extracellularly released from the microorganism. Enzymes can be alkane dehydrogenase, cytochrome P450, Weir Wigler monooxygenases, alcohol dehydrogenases, and aldehyde dehydrogenase. These are the enzymes can first add the functional group to the polypropylene or polyethylene surfaces. As a result, their hydrophilicity increases by the microorganism. And once this is hydrophilicity increases or functional group attaches to the carbon-carbon chain, then under second step is the hydrolysis of this hydroxylated polyethylene or polypropylene. So, first step is to hydroxylate the polypropylene or polyethylene surface by the microorganism by acting the different enzymes, the list of enzymes, and after then it gets converted into the simpler or monoform which can be utilized by the cells for their growth and metabolism. So, hydroxylases, which are different kind of hydroxylases present that can facilitate particularly for conversion of carbon-carbon chain, converts the functional group, add the hydroxyl group there to reduce the hydrophobicity is the first step for the degradation process, and followed by then the another hydrolytic enzyme acts on the modified chain. So, these are the list of enzymes have been reported that used to the polyethylene degradation or polypropylene degradation. I told these are hydrolytic enzymes such as cutinase, lipase, esterase, which has been reported in the different type of microorganism that extracellular releases can form the carbon ketone hydroxyl bond and some other bonds for the helping in the degradation of polyethylene or polypropylene. So, I have taken the one example of this particular strain which synthesize the P450 enzyme, and P450 is the oxygenases that can form the ketone in the hydroxyl group and also involved in the amide bond formation. So, this particular bacteria, so there is a report they have grown this bacteria in the different concentration of polyethylene. So, this is that they have measured the growth of this particular bacteria, Bacillus thuringiensis JNU1, that degrade the polyethylene. So what they did, they grown this bacteria in the different concentration of polyethylene, and if you look at the growth, this as the concentration is increases of the substrate, the more amount of growth is observed. So that means this Bacillus thuringiensis JNU1 strain tends to degrade the polyethylene, utilize the polyethylene for the growth. As a result, the growth is increasing. You can easily visualize the turbidity of the bacteria grown into the agar medium. In that media has the polyethylene present in this agar. So this particular strain of the bacteria, which is JNU1, can also decompose the polyethylene into the low carbon monomers. So this is the GC mass spectro diagram, which you can see the different small molecules. This is decomposed the small molecules with the treatment of Bacillus thuringiensis JNU1, that can show that the polyethylene can degrade it into the different type of small carbon molecules. They have also measured the, so once the plastic is adhered, once the microorganism adhere to the plastic, it increase the hydrophilicity. If it is the hydrophilicity is increases, the water contact angle gets water contact angle reduces. So they have what they have measured in this particular area. This is the non-treated polyethylene film, and they measure the water contact angle, which is around 97 degrees. And once they have treated the same material with the Bacillus thuringiensis JNU1, then the water contact angle gets reduced by from 97 to the 77. It means there is the hydrophobicity of the polyethylene gets decreases. As a result, the water contact angle is decreases with the action of this Bacillus thuringiensis JNU1. They also measure this the nature of action of this Bacillus thuringiensis JNU1 strain onto the. So this is the polyethylene sheets, and this is the treated sheets. They have observed the different magnifications. So when they have treated this polyethylene sheet with the Bacillus thuringiensis, the morphology or the structure of this polyethylene film gets disturbed. It means the polyethylene sheets gets be degraded or utilized by this Bacillus thuringiensis JNU1 strain. Another, they have find out the role, what are the properties of this particular bacteria play to degrade this polyethylene. So they identify there is a cytochrome P450, which is the hydroxylate the polyethylene. I told when in case of the polyethylene degradation process, first microbacterium binds attached to the plastic surface, and it releases some enzymes such as this cytochrome P450, which hydroxylate the polyethylene, and in hydroxylate means OH group is attached to the carbon-carbon backbone. So what this particular cytochrome P450 is involved to the degradation of polyethylene. So they have measured the different type. These all three are the different type of cytochrome P450, but as compared to this and this, the activity of this cytochrome P450 102A5 is the highest activity in presence of this. It means this particular cytochrome P450 hydroxylate the polyethylene by this Bacillus thuringiensis JNU1. And the same thing they have also measured by the GCMS. So they did the GCMS in absence or presence of this enzymes treated with the polyethylene. So this is the treated polyethylene with this cytochrome P450 gets some alteration in this, which is the sign of the hydroxyl group attached, or hydroxyl group emerge into the polyethylene. Next research is related to that can degrade the polypropylene, not polyethylene. So this is the bacteria Pseudomonas aeruginosa WGF6 efficiently degrade the polypropylene. So they have checked another research, they have checked the growth curve of this Pseudomonas aeruginosa WGH6 in presence or absence of the polyethylene. So this is the minimum media and the polypropylene with this particular bacteria, and this is the minimal media with polypropylene with this bacteria WCS6. So as compared to this, WCS6 has the higher growth, although the WZH4 is also grow in the presence of the polypropylene. And next one, this is the only the MS medium with polypropylene, and this is the MS medium without substrate polypropylene. So this particular graph shows that this WGH6 and WZH4 grew in presence of the polypropylene, suggesting that polypropylene getting utilized by the Pseudomonas aeruginosa cells. But as compared to 461 is more efficient, degrade or utilize the polypropylene during this growth. The same they have also measured with the weight loss of the polypropylene. So this is the WZH4 and this is WGH6 of the aeruginosa, Pseudomonas aeruginosa. So as compared to WZH4, WGH6 having approximately 70 to 18% polypropylene weight loss observed in this case, and also the water contact angle is also reduced in case of the WGH6, which is 56, in that case is 76 degrees. So it means this W6GH6 of the Pseudomonas aeruginosa efficiently utilize or degrade the polypropylene for their growth, bind it and degrade it into the simpler molecules which can be metabolized by this particular bacterial strain. Next one is PBC, which is very recalcitrant to degrade by the microbes. Very few reports are available, only 1 or 2 reports where the microorganism can degrade the PBC. So this is the polyvinyl chloride, and there are different kind of enzymes are involved, and this PBC can convert it into their different monomers like alkanols or alkanoic acid. So what are the mechanisms? So degradation of the polyvinyl chloride is started into 4 different steps. First step to removal of chlorine, that is oxidative dechlorination is the first step. So the bacteria adhere to the PBC surfaces and tends to release some enzyme that can dechlorinate the PBC, and that forms the dechlorinated PBC. There are a number of enzymes such as non-heme chloroperoxidase is released by the bacteria, and this particular enzyme catalyzes halogenation using hydrogen peroxide. Also the perhydrolase is also enzyme releases, which per acid forms from acid or H2O to hydrogen peroxide, and acid can form the peroxide. That is the function of the peroxide perhydrolase. Halo acid dehydrogenase is also used, that cleaves this particular bond, CX bond, CCl bond, and releases the halogens and form the hydroxyl group, OH group it achieves. So first step for the degradation of PBC is to oxidative dechlorination, in the removal of chlorine first, so that can be the chlorine can be present in the position, chlorine can be hydroxyl group, ultimately tends to reduce the hydrophobicity of the PBC. Second step of this is oxygenated PBC rather than I mean that nature of oxygenated oxide group is increases, that increases the hydrophilicity of the PBC, can make a vulnerable for the degradation. So there are different type of enzymes can be induces, like induces hydrolyzable functional group formation, and these are the list of enzyme can be used that increases the hydrophilicity by adding a more oxygenated group, more oxide group to the PBC chains. Third step of this is hydrolysis or fragmentation. After the PBC can be oxygenated, having a more oxygen group, then enzyme can easily attach, because the nature of PBC gets relatively more hydrophilic. And the next step is the hydrolysis of the PBC material. So the function of this, the depolymerize oxygenated PBC, and in that case different type of hydrolysis can act onto the onto the oxygenated PVC like esterase, dehydroxy acid, dehydrogenases and lipases. These are the list of enzymes can be used to hydrolyze, convert the PVC into the simpler molecule. And the next one, once the simple molecule gets synthesized, can be used, can be metabolized by the bacteria. So these PVC degrades in the 4 different steps. Different type of enzymes are involved that can first convert the most hydrophobic or recalcitrant this PVC into the hydrophilic nature and then increase the hydrophilicity. Once the hydrophilicity is increased, then easily hydrolyzable enzyme can act on that to degrade the PVC into the simpler molecules. So this is the one report, one bacteria, this is the only report I got for PVC degradation, that is Klebsiella EMBL1, that can efficiently degrade the polyvinyl chloride. So this is the control cells, which is the polyvinyl that in the medium, and this is the bacteria grown, the Klebsiella, that can disturb the structure of PVC when it is grown onto this surface. So this particular strain can efficiently lose the weight. They have measured the weight, but this is the control using a different time period study. The time dependent, they have also grown this particular Klebsiella strain. So with respect to the time, the approximate 25% loss in the PVC was observed at around 90 days of incubation of these Klebsiella strains. They have also measured the degradation of PVC in presence of the Klebsiella EMBL strain, and they did the GCMS. So they found that the polyvinyl chloride gets degraded into the small molecules, small carbon molecules such as this. One is this nanodol adipic acid. This is 2 is the another adipic acid, methyl osteoesters, and this octyl ministate is this is the compound, and so other compound was also reported. As a result, this long chain PVC can convert it into the simpler 6 to 8 carbon long compounds by the action of Klebsiella EMBL01. So this is the overall how Klebsiella act on the PVC. So this is the PVC that can oxidation. First the dechlorination, the chlorine group is removed, and then the depolymerization, then deoxygenases, and ultimately is formed the simple molecules that can enter. Then also different type of leakage, monooxygenases, esterases active. Ultimately this PVC is converted into the acetate, acetic acid, and some other molecule. The acetate can be utilized by the bacteria for their metabolic activity. And another way, the monooxygenase, dioxygenase also form the fumarate and some other molecule that can enter into the PCA cycle. So the PVC molecules can be degraded into the small molecules like fumarate or acetate that can be utilized by the bacteria for their metabolic and the growth purpose. Next, last one is the degradation of polystyrene. So the polystyrene mechanism can be the polystyrene is the repeated chain of the styrene. So this can be cut during the degradation mechanism. The polystyrene has to convert into the styrene monomer. So this is the styrene monomer, and different type of enzymes can be used to cleave this polystyrene chain to this form this styrene, aromatic group in the styrene, and then ultimately the styrene converts into the acetyl coenzyme or acetyl A, which can enter into the cell for the metabolism, and acetyl CoA participate into the PCA cycle. So this is the degradation mechanism of polystyrene, in which the multiple enzymes are involved like lipase, esterase, peroxidase, P450 oxygenases that can hydroxylate the polystyrene chain for the degradation. And this is the one of the research is Pseudomonas aeruginosa DSM-15071. This particular strain they have isolated from insect larvae, and they grown this strain in the film of the polystyrene. This is the control film. This is the microorganism grown on the polystyrene film, and this white is the growth of the Pseudomonas species. It means this Pseudomonas is happily growing in the presence of the polystyrene in the minimal media. They have further observed that this is the SEM diagram, this is the control, and this is the Pseudomonas species. So with respect to the time, this as the bacteria grown onto the polystyrene surface tends to change their surface, make the surface more rough. I mean the polystyrene gets utilized by this. Also the serine hydrolysis, they find out how the polystyrene gets degraded. So they have measured the different gene expression of different genes such as alpha beta hydrolases, esterases, hydrolases SCT, and also measure the SH serine hydrolases. So they find out that as compared to the other tested genes, the gene expression level in presence of the Pseudomonas treated cells having the higher serine hydrolases that helps to catalyze the polystyrene by this Pseudomonas aeruginosa. And to validate this result, they have used this SH inhibitor, serine inhibitor, with using two different media, nutrient media and this polystyrene containing media. So this is the control in which there is no effect of the inhibitor. As this concentration of inhibitor increases, the growth is also increasing in the nutrient medium. Once the cells were grown into the minimal medium having the polystyrene, in presence of the inhibitor, as the inhibitor concentration is increases, the growth of the media is also decreases. It means that SH group is involved in the polystyrene utilization by this strain, Pseudomonas aeruginosa. And this is the different research which can tell you that proteinase K induces the biofilm formation of polystyrene. So in this particular case, there is a bacteria Rhodococcus ruber 208. They have grown onto the polystyrene surface, and then the bacteria biofilm is treated with different type of proteinases, like they have treated with proteinase K and also treated with trypsin. So as compared to the trypsin, when this bacteria biofilm is treated with the trypsin, the biofilm gets disturbed, while in case of this proteinase K, treated this biofilm of the bacteria with the proteinase K, the biofilm gets more stable, multi-layered. So this is because of the proteinase K degrade the detachment signaling and leading to form the multi-layered, non-dispersed biofilm as compared to this. And ultimately, they have also concluded there are certain kind of enzymes that can be inhibited by the trypsin. As a result, the detachment is occurs. And in this case, it induces some kind of signals, can forms the biofilm related, can reduce the quorum can induce the quorum sensing. As a result, the more amount of biofilm, but the nature of biofilm gets changed. In this case, the nature is multi-layer biofilm is formed by the activation of the proteinase treatment of Rhodococcus rubers. So these are the different enzymes involved in the polystyrene synthesized in the different organisms like lipase, esterase, hydrogenase, peroxidase that can be reported in different organisms. And the last one is, so far we have discussed the single particular organism, natural condition, degrading the different type of plastic. So they have modulated the strain to increase the plastic degradation efficiently. For that, they have generated some strain. So they have used the recombinant DNA technology approach to clone the more than one plastic degrading gene into the vector, and then such vector transform into the E. coli BL21D3, and that recombinant host can be used for the measurement of the plastic degradation. So this is the one example they have measured to plastic degradation. In that case, this is the empty vector, which is empty vehicle, and in one case they have cloned this DGC genes. This DGC is the diagonalate cyclase, and this particular gene increased the cyclic di-GMP, which increased the biofilm formation. So in this one case they have make such kind of clone. Another case they have also develop a clone this WSPR genes, And third case, they have also make a single gene clone into this factor DH3. So DH3 is the PAT degrading enzyme, polyethylene terephthalate enzymes, and also TF-PUT2. They have also cloned this TF-PUT2 gene into the vector. Single as well as the double cloning they have made, and also check the polychlorolactone label in this all kind of recombinant strains. So as compared to this DH3 single clone gene, this double gene having the PH3 and DGC gene having the more efficiently degrade this PCL, and if it is WSPR gene is cloned along with the DHR1 has the lower degradation capacity as compared to this. While this TF-PUT2 and WS are the highest one, and this CUF2 and DHC has these two has the higher capacity to degrade the polyactic PCL more efficiently up to 80%. So rather than using a single or natural approach, the recombinant approach is the more efficiently degrade the plastic up to 70 to 80% if we clone multiple genes, and such recombinant host can be efficiently degrade the plastic. So rather we can use the natural as well as can also develop a genetic engineering into the strain for efficient degradation. So if we conclude the overall study, we can say that there are different type of plastics are available in the ecosystem, and the different microorganisms attack onto the plastic for their degradation. In the plastic degradation process, microorganism adhere to the surface, tends to decrease the hydrophobicity of the plastics, converts into by changing some functional group hydroxylate, and also further increase the hydrophilicity. Such kind of modification occurs in the different type of plastic, makes vulnerable for the degradation by the action of different hydrolytic enzymes secreted or synthesized by the microorganism biofilm or extracellular release from the microorganism. These are the conclusion of overall slides. These are the bacteria which acts in the different type of plastic, produce the different type of hydrolysis enzyme, oxygenases enzyme, proteases that ultimately convert the polymers of the plastic into the monomers, and these monomers can be utilized by the different type of microorganisms for their growth and metabolism. Okay, thank you.
Good morning, sir. Will you please differentiate between the thermosetting plastic and thermoplastic one more time?
Bolo.
Will you please differentiate between the thermosetting plastic and thermoplastic one more time?
Thermosetting plastic are those which we can heat one time and it gets set, cannot be remolded. It is hard substance.
Okay.
While thermoplastic we can heat it and then again we can heat it, then again it can be molded.
Okay, sir. Thank you.
This is the difference.
Good morning, sir. So my question is, let's suppose there is a persistent plastic biofilm. So sir, can it evolve to a synthetic biome?
Synthetic?
Biome.
Yeah, we can do this one. Even we can also develop different type of nick we can also generate. Consortium we can also generate for efficient degradation. That we can do.
And sir, is there any method of plastic degradation by this biofilm being sustainable?
So far, if we look at this last slide I have given, if we use this multiple organism or multiple clone that can efficiently degrade the plastic, that can be a sustainable approach. But we need to look at whether that approach, how long it will work in the natural environment, whether the plastic, how efficiently it degrades. Because sometimes if we grow the microorganism, in the natural conditions, the growth of the microorganism can also be suppressed. So depending upon the environmental condition, and the plastic nature, the growth may hamper. So, for the sustainable degradation of plastic, we can use in the control condition rather than to the natural condition. Or maybe that not today, but later on the microorganism may also develop such kind of nature, such kind of alteration that may efficiently degrade the plastic. If you look at the history of back like 20 years back or 25 years back, as such no microorganism present in the ecosystem because the plastic level was less. But as that ecosystem has the more plastic, so microorganism tends to develop their own adaptation against the plastic can efficiently degrade. It may be in future the organism can be efficiently emerge depending upon the situation of the plastic.
And sir, one more question is:
Yeah.
Is there any such species by removing which it can hamper the degradation process?
Removing the degradation process?
No, sir. Is there any keystone species in this method?
Keystone species?
Yeah. After removal of which this process will get hampered.
So, after certain, it normally in this case, some other case as well, if it is the higher concentration of simple or end product will be higher, then the overall growth of the microorganism can also be suppressed. So, there are two factors, one is the degrading mechanism, another one is the how much amount the last or end product is synthesized, whether end product is suppressing the growth or not, that we need to take care about the degradation process.
Thank you, sir.
In Immunity, Journal of Clinical Microbiology, and many others. She has won many awards to her credit, like DAE's Outstanding Investigator Award, National Bioscience Award from DBT, NASI Reliance Industry Platinum Jubilee Award. She is Fellow of National Academy of Science and Indian National Science Academies, and many more. So her CV is quite long. So just to be brief, I have put up the brief. Her lab is mainly working on model system of Salmonella, that is one of the gram-negative pathogen. Its pathogenicity, quorum sensing mechanisms within the Salmonella species. Her laboratory is working using wide range of host spanning cell lines to higher eukaryotes like C. elegans model system and Rhabdosis species, and mice to understand the host pathogen interaction. And for Professor Deepshikha, I just introduced about the audience that we are running this GIAN program, which has been sponsored from Ministry of Education, where Professor Arun K. Kumar Bhunia from Purdue University is the course instructor, and I'm the course coordinator. We have 56 registered participants in this program from different universities across, from Bengal also, from Uttar Pradesh, from Haryana, and many others. And the audience includes UG, PG, and PhD scholars. So from the last 5 days, we were having lectures from Professor Bhunia and other university faculty members, and they have been introduced about the biofilms and the microbes, because they are from the different backgrounds. Some of them are from the zoology, some of them are from like vocational studies of this medical laboratory technology. So we have a wide range of audience. So I requested you also that about the audience, that we have a mixture of audience. It's not pure microbiology students, they are from different branches. So now I request Professor Deepshikha to take over the dice, and it is an online. It would be have very better if you could come here in the physical mode. But as per your commitments, so okay, thank you for joining this online session, and over to you then.
So thank you, Dr. Gunjan Goel. It had been a pleasure to interact with you. Can you all hear my sound? Is it clear? Is it clear? Am I audible? Okay, great. So thanks, Dr. Goel, for communicating, and I'm very sorry as I couldn't make it in person. So because these kind of lecture series are extremely important, which we conduct, and only one lecture is not sufficient. If these lecture series, if we really want, I want you to introduce to the world of microbes. So at least 7 to 8 lectures are important to convince you that what microbes are. But at an outset, let me tell you that today we will learn something which may be very important to all of you who are in a field of microbiology, or who are in a field of medical lab technology, or in other branches of life sciences. That is a community behavior in bacteria. Some myths will be bursted, saying that microbes are unicellular. They are, of course, but they don't work in a unicellular fashion. So let me share my slides, and kindly make sure to ask me questions whenever you would like to feel like, so that we can get our work cleared, or the doubts cleared. Is my slide visible? Is my slide visible? My slide is visible? Okay, great. Thank you. One of you, if I am not audible, if my slide is not visible, kindly make sure to give me one buzz so that I can stop and then start again. Let me put my pointer. Yeah, thank you so much. So let me introduce you all to the world of bacterial communication, that is a quorum sensing. Now, at the end of the talk, I want you to be convinced and also have a very strong take-home message that when you work with a bacteria, you have to be very careful about certain features. And those features are maintaining the community structure of bacteria, and how everything can fail, right from a very simple growth curve to getting your products out in a genetically modified cell, or to make your electrocompetent cell. How the language is very important. So I am addressing my young colleagues, so I would love to have a lot of questions from them, because without the questions, any kind of the talk is a very futile exercise. So kindly make sure either to stop me in between and ask a question, or you get your questions and then we will have them all discussed in the end of the talk. Right. So there are 3 major points that occurs. Why you want to study? What is actually quorum sensing? Why one should be interested in quorum sensing, and what do we know about its history? So at the base of it, what is this quorum sensing? Now, in a textbook, you might have learned that bacteria grows by binary fission, and there is something called as symmetric bacterial growth. Of course, this also you must take it with a pinch of salt, because the symmetric division of the bacteria, that is a 1 micron bacteria getting divided into 2 daughter cells of 1 micron each, is true maybe in a classical cell like E. coli, but the asymmetric division, that is a bacteria giving rise to a long cell and a short cell, is very evident nowadays. So you must keep in mind that it is not a very strict rule of the symmetric division. But anyway, let us go by the symmetric division of bacteria. So 1 bacterium divides into 2 bacterium, then you give them nutrients, they divide into 2 bacterium, and then 2 into 4, 4 into 8, and then what you get is some kind of a growth phase. So when you draw a growth phase, you start with the lag phase, then it go to the log phase and a stationary phase. For a very long time, until now in the books, what is mentioned is the stationary phase is a result of lack of nutrients. That means the entire nutrients has been exhausted. But then, you must think that with a variety and enormous source of nutrients that you may give, is it really only the nutrients that is making the bacteria to the stationary phase? And the answer is no. It is not only because of the depletion of the nutrients. It is because that when the bacteria are enough in numbers, they give the signals. Now, this signal is a way for the bacteria to count their numbers. That is, we are good enough among ourselves. This particular number is known as a quorum. That means the quorum has been achieved. There is no more growth required for the community function to begin. It is something like this, as simple analogy as like, suppose in your class, you are, say, I can see in the audience, say maybe 50 students. So 50 of you has been registered. So your quorum is 50. It cannot be 51, or it cannot be 1 or 5 either. So it is that number. Now, the question that is actually bothering all of us in the field of microbiology, including infectious disease, is how the bacteria understands their number. Because number is everything. Be it a disease, or be it making enzymes, or be it making biofilm. It is a number which is important. If the number is wrong, or if the number is perturbed, none of these functions can happen. So it is about the number. Now, how this number is being achieved is a question that is, in fact, still perturbing in the sense of or instigating our thinking to get in the direction of understanding the bacterial communication. That means, do the bacteria have a language? So what are these signals? So we will try to understand how bacteria know their numbers, that they are enough now to start the processes. What are these signals? Now, it is not that the bacteria has started communicating now or a decade ago. It is only that we were not geared up enough to understand the communication. It has been done from time immemorial. The origin of the earth 4.6 billion years ago, just a billion years later, bacteria originated, and from then on, it has been doing this. But we were very late in introducing the bacterial communication. And that is why this research is at the helm of affairs. It is at the top of the hierarchy if you really want to understand the bacterial physiology and the bacterial life. Now, what are these signals? One of the signals that we will discuss today, they are known as an auto-inducer. Now, why are they known as an auto-inducer, and how does it play an important role? These auto-inducers, this one of these molecule, is nothing but an acylated homoserine lactone. It is made by the bacteria and it can be exported. Now, whenever anything that is made by a community of a bacteria and is given out as a public good in the environment, that means the bacteria is sharing the resources. Yeah, it is not keeping the resources to itself. So the bacteria is giving it out as a public good. And this public good, that is this AI molecule or the auto-inducer molecule, can be used by the other bacteria. So this, in fact, brings us to something called as a division of labor in a bacterial community. Now, suppose in a million of bacterial community, there is a proportion of the bacteria that makes this quorum sensing molecule. There are other bacteria who can take up this quorum sensing molecule and do their function. So that is the reason it is called as an auto-inducer. That means this is produced by the same community of bacteria and used by the same community of the bacteria in the system, in a given system. So, and this molecule that you are seeing here is nothing but acylated homoserine lactone, the first auto-inducer molecule that was discovered, and that became a kind of a landmark event in understanding the bacterial communication or the language between the different bacteria. So once the bacterium starts dividing and they get their number, they start producing this auto-inducer molecule. And when do they know that their number is enough and they will not grow any further? When the concentration of these auto-inducer molecule, extracellular concentration, is equivalent or to the intracellular concentration. That means when they have reached an equilibrium. At that point in time, they know that there is no longer any need to make a new AI molecule, and now they are in the quorum, the correct quorum. That means the correct quorum is achieved. Now, the word quorum has not been used in microbiology since very late. The word quorum is used in a parliament. It's a word in English dictionary. If you look, what is the meaning of the word quorum? Quorum is used in a parliament, saying that, okay, now the quorum is complete. Let us start the proceeding. The word quorum in microbiology or in a microbial community is extremely late. Only way back in 1992 was the word quorum used. So this signaling, which is called as an auto-inducer signaling, allows the bacteria to sense that their number is enough, and then they have reached a quorum. And in totality, this is known as a quorum sensing. Now, quorum sensing is an extremely explorative field. Many of you may be in the BSc or MSc and starting to look for a project. The kind of projects that you can actually think of very innovatively in this field is mind-boggling. And not only it is very exploratory, but you can do it in a very simple mechanism, as simple as a bacterial growth curve. If I am to tell you that it is as not simple as we think of, and many of you, when you do your practical experiments, you might have realized that it is not always that you get your lag, log, and the stationary phase very correctly. Have you ever questioned that why am I not getting that? Have you ever questioned that why, in spite of doing every step correct, my electroporation of E. coli to transform the plasmids inside is not happening? Why there is so much of a failure? This all is because your quorum is wrong. So if you start with a wrong quorum, the further processes of everything in bacteria will simply collapse. That is the only reason that why you should be very much learning this process of bacterial communication. So so this is called as a quorum sensing. Now let us look into its very interesting history. Now I'm sure that as a kid, when you were a kid, you might have read this book, 20,000 Leagues Under the Sea. In this book of 20,000 Leagues Under the Sea, there is something called as a Tale of Milky Seas. I think it is a chapter 2021. I don't remember because I've read it very long time back. So this Tale of Milky Seas is not a sci-fi story, or it is just not a some kind of a made-up story. It is a true event where when the sailors, they were sailing in the cruise, they realized that there is a 250 kilometer stretch in the Indian Ocean, which was glowing. Now, NASA pictures over a period of time, over years, decades together, have seen this blue stretch. Now, what is this blue stretch was the event. And this was a hallmark beginning to understand that why there should be this blue stretch. And what is this blue stretch? And it is not some 1 kilometer, 2 kilometers, 250 kilometer long blue stretch. What is this blue stretch doing? So the sailors, collected some sample from there. They were also quite, intelligent enough to collect the seawater because these bacteria, whatever the creature is, should be surviving in this seawater. And they brought it back to the lab. And from then on, it was an eye-opener to understand that why there is this blue stretch. Now, this blue stretch was due to a bacteria which is called as a Vibrio harvei. It is a Vibrio species, like you might have heard about Vibrio cholera, which is a causative agent of cholera. Like that, it is a Vibrio harvei. It is a harmless species of Vibrio, and it gives blue light. Now, the question is, what benefit it has to the bacteria Vibrio harvei to give up this blue light? And this Vibrio harvei is a free living bacteria. It is not living in association with anything. So what is this that is making the stretch, first of all? How this stretch can be so sturdy that in spite of waves and etc., it is not drifted? And what is it that gives it a blue light? Now, this glowing organism then on became a hallmark of understanding the bacterial community which glows. And sky is the limit now. There are a lot of these marine organisms where the new bacteria are being isolated, which can glow. The question is to find out the mechanism of how do they glow. So in fact, this is the history of a tale of milky seas, which paved the way to figure out that how the bacteria can glow and how they can make a glowing bacteria, and how the quorum is important for that. Now, if you define quorum sensing, it will be a simple definition of regulation of the gene expression in response to fluctuation in the population density. How? Because even in that big stretch, there are events going on, right? There is a lag phase, log phase, stationary phase. Bacteria die because they age. I am not very sure how much you are aware of the concept of aging in a bacterial population. Just because you take a loopful and always put it in the broth and they grow does not mean bacteria are immortal. So there is something called as a process of aging. Just like we age, just like the universe ages, bacteria also ages. So the fluctuations will continue to happen, and the regulation of the gene expression in response to the fluctuation and the number can be defined as a quorum, or in a very simple way, it is a way for a bacteria to count their numbers. That this number is the number which is important for the function. Now let me make you understand this in a very simple way. Like we are communicating with each other in English because we understand each other in that language, the communication between the bacterial cell is of utmost importance for the bacteria to function. How are we functioning now? I am communicating with you. You are perceiving certain knowledge in your head. And then when we communicate and understand each other, you will have a lot of questions which we will discuss. So this entire process is a attribute to multicellular organization level. That means many of us are making a kind of a society, and that is how we are functioning. It's the same thing with the bacteria. So bacteria, if they stop this communication with each other, they stop the functioning. And no doubt that why these quorum sensing molecules have become a multi-billion-dollar project. So $1 billion project, because if the bacteria cannot communicate, they will not be able to function at all. Think of it in terms of tackling an infectious disease. If you stop the communication with among them in causing infection, they will not be able to cause infection. So quorum hunters, they are called as the quorum sensing hunters or the quorum hunters. The quorum molecule hunters and quenchers, both of them are playing a very big role in pharmaceutical industries now. Now, only in the late 70s, it was understood that the bacteria communicate. The eukaryotic biologists, they did not like the idea. They always thought that bacteria are prokaryote, but I must remind you that we began with bacteria. The universe began with bacteria. We are alive, or any life form is alive because of the bacteria. We carry majority of the bacteria in our gut and in other places, except few organs. So if they are gone, entire life form will be perished. So we must not forget that we are alive today only because of them. So communication became very late. Only in 1920s it was somehow thought of, but only in 1970s it was understood that there is a communication. So the system in which, as I was discussing, that was discovered was a Vibrio Harvey. So what they did was they bought the bacteria and they plated it in a seawater medium. So get the seawater. This is a smart strategy, you see. You take a seawater, you filter, sterilize it, and then put some agar. You get your seawater medium. And then you plate that bacteria under the UV light. This emits this blue light. So these are the glowing bacteria. And also it was discovered in a very deep sea fish. This is known as a Vibrio fishery. So why in the very deep sea where sunlight cannot reach, these kind of glowing fish, glowing squid, they were observed. And this one is an excellent example of a symbiotic relationship. That means these fish with a glowing head, it is just like a coal mine worker getting down into the coal mine. They have their bulb only in the front, if you might have noticed, not anywhere else in the body. This is because to show the way. So similarly, this glowing head allows some kind of a prey to be attracted towards this fish so that the fish can survive. And the bacteria derives the nutrition from the region of the head. So they survive. So this is an amazing example of a symbiotic relationship of a Vibrio fishery with the fish. Now, the scientists have also done the experiment, though they are difficult experiments to do, in very deep, mimicking this deep sea aquarium-like condition, where no sunlight will reach or no light source will reach. And they cured of this fish of this particular Vibrio fishery. That means they killed this bacteria with antibiotic. And they injected this bacteria into other region like tail region or fin region. And they figured out that the bacteria could not survive, neither the fish could survive because the fish died. So this fishery is an important example of how the evolution happened over a period of time for the very deep sea creatures. So these two, Vibrio harvey and Vibrio fishery. Vibrio harvey is a free living organism so that it does not need any host to grow, whereas Vibrio fishery requires the fish for its growth. So in these two systems, in fact, the experimentation began to understand the quorum sensing. Now, if you look into the history of quorum sensing, it was in 1965, Thomas realized that Streptococcus pneumoniae, this bacteria is quite important for historical reason. Now, why is it important for historical reason? Frederick Griffith proved that DNA is the hereditary material by using Streptococcus pneumoniae. At that time, it was not known that how Streptococcus pneumoniae can easily take up the nucleic acid. Unlike E. coli, you don't have to put effort. You don't have to make them electrocompetent by doing many, many steps. You put some nucleic acid, they take it up. The similar is the case with Helicobacter pylori. They also take up many different kind of nucleic acids very easily. So Thomas have given it a name of competence factor. Competence factor is the factor that he named, that this bacteria, after it reaches a certain density, makes this factor which make the bacteria naturally competent. That means they will be able to take up the DNA. So he understood this and he gave it a name of competence factor. Then 5 years down the line, Nielsen, who was a chemist by training, he understood that there is a certain light produced in this Vibrio fishery, which is controlled by the community. That means it is controlled by the self-produced signal. The same self-produced signal is termed as an auto-inducer that I was talking about, acylated homoserine lactone. And then another 3 years down the line, Eberhard had identified that what this signal is, and that particular signal is none other than N3-oxohexanoyl homoserine lactone. So as I always tell to my undergrads, that the demarcation in science must be removed completely. You cannot do a good microbiology if you do not have an idea about the chemistry, physics, and mathematics. Microbiology is all about numbers. So if you have a fear of number, then doing microbiology can become boring. Similarly with chemistry, if you're very strong with chemistry, all the molecules that the bacteria produces, and they are the resources, will become very easily available because of your knowledge in chemistry. That is how Eberhard could figure out that what this molecule is. Only when this molecule was figured out, now Sigma is synthesizing these molecules. Life has become very simple. You can, in fact, buy this homoserine lactone molecule ready-made in a bottle as a quorum sensing molecule and do your experiments. Many different circuits, many different things. And then 2 years down the line, Engelbrecht, who was a genetic engineer, he cloned the gene and he termed that gene as a lux gene. As the intensity of light, unit of the intensity, lux, he honored that by giving it as a lux gene because it produces the light. You see, it is 1983, till now the word quorum has not come. Then in 1994, only in 1994, Fuqua et al., he introduced the term quorum sensing to describe the cell-cell signaling. That late the term came, quorum sensing. And homologs of the Lux eye were discovered in different species. So Vibrio fishery became an example. Now, let me also tell you, colleagues, it is not that the bacteria which produces light only do quorum sensing. This is just one small example because you can see the light production and you can do many experiments. Every single bacterium, including E. coli, communicates with each other. So it is up to us to understand that what is that sensing molecule. Okay, so quorum sensing is done by every single bacterium, and it is up to us to understand how do we go about characterizing the AI molecule, or how do we go about understanding the gene cassette, which will be important for this particular signaling. But you can take an example of Vibrio fishery where Luxi Lux R became a very important signaling communication. So molecule is very simple. This is an acylated homoserine lactone. Look at this acyl ring, and R is the side chain. So what differs in these different bacteria, this is produced mostly by the gram-negative bacteria, okay? Now, gram-positive bacteria have a very different quorum sensing molecule. They don't produce this acylated homoserine lactone. It is very different. I will come to that. So the R is the acyl side chain, and the diversity is in the acyl side chain. So the bacteria will make this AI molecule, it will send it as a public good. The other bacteria in a community will take it up, and equilibrium will reach when the extracellular concentration will be equal to the intracellular concentration. And then the bacteria knows that the quorum is achieved. It will stop the growth. Once it stops the growth, what does it do? What are the functions of the bacteria? So look into the specificity. So you see, this is a acylated homoserine lactone, same lactone ring all over. Vibrio fisheries acyl side chain is very different from the Pseudomonas aeruginosa acyl side chain. Pseudomonas is becoming an amazing bacteria to work with, not only because it is a high priority pathogen. As per the WHO, it is a top pathogen belonging to the ESCAPE group, ESKAPE. P stands for Pseudomonas there. It has a two quorum sensing system. One is a LAS-I and one is a RH2, and they're hierarchical in nature. Yeah. So see, Agrobacter tumefaciens, a different acyl side chain, Vibrio harvey, a different acyl side chain. So what is the specificity? Different bacteria will have a different acyl side chain, but the lactone ring is common to all. Yeah. So the modification of the side chain will distinguish. So it is up to you, you know, how good you are with your chemistry and with your mass spec, and you come upon a new AI molecule. And it's always a discovery phase when it comes to quorum sensing. That's why I told that it is a very exploratory, phase in bacterial communication at this moment. So what does it do? That is most important. Okay, they have made a community. What does it do after it makes a community? So this is a very small example that I have given you. Like Vibrio fishery, they make light. So there is a light production. Aeromonas hydrophila, they will make protease. So all the products that the bacteria make is a result of their number, is a result of their communication and the quorum. So this is just an example of a multicellular organism. Cell in a Petri plate will do just a cellular function. Then they will become a tissue. Tissue will become an organ, and then they perform the function as an organ. Similarly, a single bacterium cannot do anything. It is the multicellularity in the bacterial community which will do the function. Be it a production of protease by Aeromonas hydrophila, or a process of conjugation in Agrobacterium tumefaciens, Burkholderia cepacia making siderophores. Now, siderophores are very beautiful molecule that are given out by the bacterial community. What they do is they go and bind to the iron and they bring it back to the bacteria, because iron is a very important element for many of the function. Chromobacterium violaceum making antibiotics. So even the production of the antibiotic is a result of the bacterial community behavior. Irvinia chrysanthemi. This is a bacteria which is very specific to the plants. It is not a human pathogen. and it is a very strong pathogen for the plant. What does it make? It makes pectinase, because plant have a strong pectin layer, and if the bacteria needs to get inside, it has to make this enzyme pectinase. So pectinase is only produced when the bacteria have reached a quorum. Similarly, Pseudomonas aeriofaciens making phenazines, so Pseudomonas aeruginosa making a biofilm. It is one example, but the biofilm is made by many bacteria. Rhizobium etli, number of nodules. So the number of nodules in a very friendly bacteria which fixes the nitrogen is because of the quorum. Pseudomonas, Yersinia pseudotuberculosis. The motility and the aggregation pattern in the bacteria is because of the quorum. This is a very small list, tip of the iceberg. List is never ending. It is up to us to add on to this list. So every process that the bacteria does, be it a growth, be it causing disease, be it fermenting your, idli dosa batter, or be it setting your curd, every single processes and work of a bacterial community is because of the language and because of the multicellular function of a bacteria as a community. So single bacterium cannot do anything. It is a community of bacteria which can do many, many things. So for gram-negative bacteria, it is acylated homoserine lactone. Yeah. Now for gram-positive bacteria, they don't make this lactone ring. What they make as a quorum-sensing molecule are the peptides. So they make peptides. So for example, look at this. These are very short stretch of the peptide, which function as auto-inducer molecule in gram-positive bacteria. Now, colleagues, I am sure that you have a very good knowledge about the classical differences between the gram-positives and the gram-negatives, right? In terms of their cell wall structure. The major difference in the gram-positive is extremely thick peptidoglycan and no outer membrane. There is no outer membrane in the gram-positive. Whereas in gram-negative, you have a very thin peptidoglycan and you have an outer membrane. That is a basic difference. And every single, supramolecular installation in bacteria depends because on this. So you can imagine why they are not making these kind of acylated homoserine lactone and only the peptides. Because gram-positive have lot of big peptide transporters, thick peptidoglycan. Anything which has to be installed in that should span across 30 to 40 layers. So they have a very nice peptide transporters and they transport this auto-inducer molecule. But there has to be one more language. That language is a universal language. And what is that universal language? That means a gram-negative should be able to talk to gram-positives. And the gram-positive should be able to talk to gram-negative. Yeah. So at the moment, our universal language is English. Each one of us have our own core language. Say, for example, I speak Marathi. It's my core language. You may not understand. So we are speaking in English. English becomes a universal language. Or I speak Bengali. It is a core language of mine. Not many can understand. We speak in a universal language. Similarly, bacteria have a universal language. And this universal language is a different quorum sensing molecule, which is known as a AI2 or auto-inducer 2. And the interesting part of this language is that it has a boron B in its center. Now, till now, the chemists are unable to find out that how the bacteria incorporates the boron in this AI molecule. Synthetic chemist cannot make this molecule because that steps of incorporation of the boron in the AI2 molecule is extremely, extremely difficult. They can figure out, logically they can figure out, but experimentally this was not possible. So how the boron gets incorporated is still a mystery. Now, Vibrio fischeri luminescence is a prototype for a sensing system. I will just give you a genetic circuit of this. This is very easy circuit, so you will be able to understand. This is controlled by two genes, LuxI and LuxR, as simple as that. LuxI will make this molecule, okay? It will be sent out. So there are exporters and importers. And then the bacteria, which are the neighboring bacteria, they are going to take up this AI molecule and it will bind to the LuxR. So LuxR is like a receiver. It will bind to the LuxR and this complex of the AI molecule along with LuxR will go and bind to the target genes and it will do the gene expression. So this is the gene arrangement of a Vibrio fishery, and this bioluminescence will happen only when the Vibrio fishery is at a certain density. It will not happen when they are in certain low numbers. It will only happen when they have reached a certain density. And the molecule that it makes, and the first molecule which Eberhard had discovered, is N3-oxohexanoyl homoserine lactone. Yeah. So you see here, so this is the arrangement of the gene. This is the luciferase, which is going to work as an enzyme to give this blue color light. The question for Vibrio fishery is simple, because Vibrio fishery required the light for the fish. Fish should hunt and they should get it. That is quite understandable. But why the light is required for Vibrio harvey, because Vibrio harvey is a free floating, is still an open question. Now you see that the Vibrio fishery circuit is very simple. Lux-I, Lux-R. You can do an amazing experiment in the lab by knocking out this Lux-I or deleting the Lux-R, and then ask a question whether you can still see the light. You can measure the light with the help of a bioluminometer or a luminometer. It's a simple instrument one can buy in the lab and measure the light formation. And you can make a genetic circuit, switch on and switch off system, by adding the quorum molecule, by deleting the quorum molecule, deleting this Lux-I which makes the quorum molecule. And as I mentioned, sky is the limit to actually think of a very innovative ideas about the experimentation here. Now let us come to Vibrio harvey. Vibrio harvey is a marine bacterium, but unlike Vibrio fishery, they are free living. They are not in any symbiotic association with anybody. But then the question came, and the marine microbiologists are trying to ask it from a very long time, how is this stretch maintained? It cannot be just bacterium. How much ever strong they are in making the biofilm, it is not possible for the 250 kilometer stretch to remain like that. So there must be something, some zoonotics, some planktonics, which are getting attracted and coming to the surface and harboring there. They may not be dependent. Vibrio harvey may not be dependent on them, but there is something more than to Vibrio harvey in that stretch. But unlike Vibrio fishery, the quorum sensing circuit is complicated in a Vibrio harvey. Let us see how complicated this is. See, in Vibrio fishery, you have that just Lux-I and the Lux-R. Vibrio harvey, you have two systems. This is the one system which makes one kind of a quorum molecule, and this is another system which makes a different kind of a quorum molecule, you know. So this is a hybrid system which is behaving both like a gram positive and as a gram negative. So what is the relay apparatus? The relay apparatus is nothing but a sensing system and a response system. So it will sense the quorum sensing molecule and then it will send the response regulator. So the Vibrio harvey secretes the AI2, and this AI2, this is the one which is designed for interspecies communication. That means it's a universal language. So this AI2 can also communicate with other gram-negative bacteria and also gram-positive bacteria. So the universal language or the other interspecies language, this AI2 is made by this Vibrio harvey. So they are very smart. They make both the AI1 and AI2. They can communicate with various different species of bacteria, and they are free living. So now the best part for us as experimental biologists is that you can bring them to your lab and you can grow them. This bacteria is being given by MTCC, that is a microbial type culture collection in IMTech Chandigarh, and anybody can buy this. It's very cheap. I think hardly it costs some 2,000 rupees per strain, or 2,000 or maximum 3,000. And then you have a simple media for them to grow. And even if you do not have a luminometer, you can at least put up an experiment and see it under the UV to see the glowing light, you know. So sky is the limit here to, in fact, get all your ideas out to work with this particular circuit with Vibrio harvey. Because it can make both the quorum sensing molecules, it can sense both the quorum sensing molecules. And there are many complications. What are the complications and what are the questions? If it can make both the AI1 and the AI2, what is the window frame, of making these molecules, because they have a very common denominator. That is a very common circuit or a transcriptional regulator. So how this transcriptional regulator is regulating both these molecules, as it is common to all. That is, a LUX-CU is a common regulator between the AI1 and the AI2. And that is how the complications is making the study far more interesting. You can make a knockout in Vibrio Harvey fairly simple by using a one-step deletion technique. And then you can ask various questions. That if you knock out this, what is the status of AI2? If you knock out AI2, what is the status of AI1? Or if you knock out both, how does the bacteria behave? How do they still determine their numbers? So this is a very important tool for all of us to study. Not only that, you can access or you can by yourself discover other quorum sensing molecules, say from unknown bacteria, and then put it on the Vibrio Harvey and ask a very simple question. Is that unknown supernatant or from other bacteria, in fact, giving the blue light? If it does, that means it has some of the quorum sensing molecule, and that is how the blue light is produced. So that is how you become a discoverer. Now let us come to Pseudomonas aeruginosa. As I mentioned, Pseudomonas aeruginosa is crossing the boundary of being only opportunistic pathogen or lying in an environment. Now, Pseudomonas aeruginosa is part of the escape pathogen, which is a high priority pathogen. And why Pseudomonas is creating so much of a complication? Let me just put a pen here so that I can make you understand. Now, this particular Pseudomonas aeruginosa's complication is because of the two systems, One is this Lassi-Lasar system and another is this RH2 system. And they are hierarchical in nature. What is meant by hierarchical? If this system is active, this system will be active. If this system is inactive, this is inactive. That means the information is passing from this to this. Okay? Now, if this system is inactive, nothing will happen to LASAR. This will be functional. So this is hierarchical. That means it is coming from LASR to the RH2. And these two hierarchical system is creating a lot of complication in the gene, in the system. Now let us understand how. Now, Pseudomonas aeruginosa can in fact create a pathogenesis. Why is it notorious? It is notorious because it can cause from a diabetic foot infection to lung infection to any kind of wound infection, septic shock, nosocomial infection. In the ICU, a very, very high death rate or the morbidity now being reported by this Pseudomonas aeruginosa. So it has become a really, really very important pathogen to tackle. And it is a pathogen one once gets in because of the high antibiotic resistance is also very difficult to clear. Okay. So what it does and what is a hallmark is it makes this AI1. Now the AI1 is being received by that is LASI is making it. It is being recognized by the LASR of the receiver, and they make certain molecules, right? Like elastasis, toxin, pyoverdine. They will communicate with the cell. They can mount an host attack. See, that is how they are coming together. Now this coming together is nothing but what you call in a layman's term a biofilm formation or a microcolony formation. Now, within the tissue, within the host, now our own lab, we worked on this in vivo biofilm in the host tissue, and you can indeed see them making many of the bacteria. We work with Salmonella and we also work with escape pathogen. We see that these kind of biofilms are being laid. So this is the community behavior. But only when the AI1 along with this LASR is there, it will trigger this second system, that is the RHR system. Now, this particular system, once it is triggered, will make a AI2 molecule, and then they will make many other virulence factors, pyocyanine, biofilm, they will mount an host attack. And the beauty of this pyocyanine, this is blue in color. I'm not sure how many of you have grown Pseudomonas aeruginosa in the lab. You will see that if your process is very correct, if the quorum is very correct, and your growth conditions are favoring the crosstalk, then your broth will turn bluish green in color. Okay, so you will see a bluish green, and the bluish green is nothing but because of the secretion of the toxin pyocyanin. So again, one of the great system to work with. If you cannot have your hold on the Vibrio harae, and you can use Pseudomonas aeruginosa, because this is in the visible range. This pyocyanin production is in the visible range. So you can use any simple spectrophotometer to get this work done. But this requires a biosafety level, and you require institutional biosafety ethics committee approval. You cannot work with Pseudomonas aeruginosa just on your bench. So all of you should be very careful, even when you're handling these pathogens. And if the care is not taken, it manifests, the disease get manifested way down the line. So you need to be very careful when you're working with these organisms. But again, this is one of the most beautiful system because of the hierarchy. So if the LASI is LASAR is functional, this is functional. If this is non-functional, the entire thing becomes non-functional. And that is how it mounts the host attack. 60% of the diabetic foot infection and the complications are because of the Pseudomonas aeruginosa and its combination with Acinetobacter baumannii. Yeah. Now, in contrast, when you look into the gram-positive bacteria, they're fairly simple. They make these kind of peptides, and small peptides as a quorum sensing molecule, and they use this ABC transporter to transport the peptide. Very simple. These peptides can be isolated, and then you can do a lot of experiments. So this is not as difficult. And how do they recognize the signal? They recognize it with this kind of a two-component system, where the signal peptide will bind to this receptor. Then it will do a autophosphorylation of a histidine moiety, and this is a relay race. Yeah, so the phosphoryl group from the histidine is transferred to the aspartate, and this is a response regulator, and then this will do the target gene expression. So this two-component system is a signaling system in bacteria. They are present in all the gram-negative, gram-positive. They are the hallmark of a signaling mechanism that is in bacteria. Just like in mammalian cell, you have a ligand and a receptor. Similarly, in the bacterial system, you have that. So very quickly, for some time, I would like you to get fascinated by the bacterial community behavior, something which is very close to the apoptosis in the mammalian cell, to make you understand that how you should be appreciate if you are a microbiologist, you are progressing towards doing your PhD or your higher studies, you're using a bacteria as a system, how you should be appreciating the every process that happens in higher eukaryotes, even in a bacteria, including the programmed cell death. The programmed cell death in a mammalian system is known as apoptosis. You do not use the word apoptosis in a mammalian system, in a bacterial system. What you call is a programmed cell death. Now, this programmed cell death is a very interesting system. This is modulated by a module called a toxin-antitoxin module. One example that I'm going to give you here is a MASI-MASF system that is in E. coli. Like this, there are many toxin-antitoxin modules. There can be many questions that will arise in your mind. Why a bacteria should have this toxin-antitoxin module to begin with? Because what is this module? This module, which is toxin, is encoded by MASF. Yeah, MASF is a toxin. This toxin is a specific endoribonuclease. That means it will go and cleave the mRNA of the bacteria. It will cleave it in an ACA sequence. It will harm, it will self-harm. But the MAS-E is an antitoxin, so they are never found free. MAS-E is always in very close association with MASF. This is the twin module system. Only when the MASF is free will it lead to havoc. But the question comes, why a bacteria to begin with should have this suicidal mode? Why? Why do you think they should have this suicidal mode of action? That when the F is set free, they are going to create this problem of cleaving its own mRNA. Now, this particular system is there in E. coli is an example, but in all other bacteria, which is a stress-induced antitoxin module. What do you mean by stress-induced toxin-antitoxin module? That means whenever any bacterial community is under stress or under threat, they will execute this. By the way, why they should execute this? Tell me, why they should? How many of you are very fond of Harry Potter? You know Harry Potter stories. I'm sure many of you are. It's like you still remember Rising from Ashes, the Phoenix effect. Bacterial community cannot be exhausted under any condition. So it's something like that. Even under stressful conditions, say antibiotic stressful condition, majority of the community will collapse. There is a population which survive, and that population comes up like a phoenix effect. And for that small population to survive, the death of the majority of the population is important, because the minority of the population, they will feed from the released sugars, amino acids from the dead population. So I'll tell you what happens and how beautiful the system is. The system was discovered by Kulka's group in Finland 2 decades now. It made a big comeback. It was published in Science and in Nature, Journal of Bacteriology. From then on, there are many twin systems came up. We ourselves published a paper in EMBO Reports 2 years back saying that indeed this system works in a diverse bacteria, including Salmonella. So whenever there is a stressful condition, there will be expression of this MAS-E/MAS-F. Look at the beauty of nature. MAS-F is a very stable. MAS-E, which is an antitoxin, this is a labile. What is meant by labile? Labile means not a heat labile or pH labile. Labile means this MAS-E gets destroyed with the help of a protease, which is called as a clip protease. Now, this clip protease is also made by the bacteria or overexpressed by the bacteria in this antibiotic stressful condition. Okay, and the clip protease will cleave this antitoxin, and that is how your toxin is set free. When the toxin is set free, what it does? It will cleave the mRNA at the ACA sequence, and as a result, there will be a population-mediated death phenomenon. And that death in a population is triggered by a quorum sensing factor, which is called as a EDF, or the extracellular death factor. Yeah? So, have you now understood the beauty and the synchrony of the process? When there is an induction of this MAS-F in any condition, especially in an antibiotic stress condition, there will be an inhibition of the large synthesis of the protein. Yeah, but here is a catch. There is a differential ability for the bacteria to induce either the survival genes or the death genes. This death and this death is not different. They both match, you know. This is a very fast death. This is a bit of a slow death. But the survival population, a small population will survive, which will feed on the dead population. And this is done by the extracellular death factor. Now you know that why we are not able to tackle the infectious disease, why the problem of antibiotic resistance are happening, and what is the reason that we should not now go on, you know, discovering newer antibiotics, because whatever you do, your toxin-antitoxin system will trigger in and it will allow the survival of a smaller population. And it will go on and on and on. So the way to tackle infectious disease is not at all by discovering a new antibiotic. We are making our own life very, very hell. You know, it is a right to live. If we have the right to live, so are they. Just because it is causing us harm, we are going on discovering. And they are in nature, they are always in association with certain other bacteria which are continuously producing them, antibiotic, right? Now, this is nothing new. They have already gained that resistance from the soil. And once they enter inside the human body, we are pumping again. So now that you know about the community behavior, you know about the programmed cell death in a bacterial community, you should be very, very careful about thinking and designing your project or having a very novel thought process. I will not take you to the experiments because the experiments are very interesting, but maybe sometime next time if I come there. So what happens is whenever there is a stressful condition, the Massey-Mazef is triggered. Stress can be any antibiotic and it is not dependent on antibiotic. So remember the antibiotic stress, it can be chloramphenicol, trimethoprim, ciprofloxacin, you name it. There will be release of this toxin. This toxin will cleave the mRNA at ACA sequence. Of course, there is certain signature. And this will lead to inhibition of the large synthesis of the protein, okay, and death of the population. But in a minuscule population, there will be an increase in the survival genes, which will lead to survival of a small population. Now, the small population is a key. This small population will feed on the nutrient which are released by the death population, and then they will grow. So now you see how a bacteria, which we thought it is so simple, it is unicellular, can be so complicated, just like any other so-called higher organisms. So our definition of the higher organism itself is wrong. Just because there is no center of so-called brain, that well-defined brain in a bacterial community, does not mean that they don't do things. They exactly do the things. They are very diverse. They are very fast. Their signaling system is within milliseconds. They can adapt to any environment and they do not perish. Under stress conditions, they do not perish. And it is all because of their community behavior. So thank you, colleagues. I am very thankful to you for having this interaction on, and I am open to questions now. So colleagues, it is very important that you should ask questions, and it can be as simple as what is bacteria, does not matter, but the questions that have come to your mind regarding the bacterial communication should come out. So this is important. And you should have your own query. If you have faced any trouble in your own experiment system, not able to grow bacteria, the bacterial growth is not happening, your antibiotic is not working, you're not seeing the zone of inhibition, many things that can happen is because of that. So thank you very much. Yes, sure. You please introduce yourself and then you ask a question. Let me know also who is asking, because it's very important. Yeah. Okay. Yeah. Thank you. Yeah. Yeah. Yeah, very good question. Can it be used as a tool? Yes, it can be. Now, the only problem with the Vibrio fishery is that Vibrio fishery, you can grow them. Nowadays, there are some kind of a media that is available for the growth. And you actually can use this light formation as a tool. How you can use it as a tool? So suppose you want to know a certain agent, say bioremediation agent or any other agent, whether it is working on a community or whether it is disturbing the quorum, you can use it on the Vibrio fishery or in a Vibrio Harvey, because Harvey also makes your blue light. Yeah. And then ask a question that now whether your agent or a molecule has stopped making the blue light. And if it does, you have your answer. That molecule X or molecule Y is acting like a quorum quencher or a disruption of a quorum. Or it can be enhancing the quorum. That means it is acting in a way by which it allows the crosstalk to happen. Yes, definitely you can use it as a tool. How is it possible that the quorum is so high and there is only one question? Not possible. Ask. Sorry? No, that is your quorum. I'm sure that they have many interesting questions in mind. It's similar to what I was talking. Yeah. Yeah. You tell me one thing. How many of you have grown E. coli? Raise your hand. How many of you have grown E. coli? Let me see. Okay. And how many of you have faced problem in getting your very characteristic lag, log, and stationary? There are, right? Now, very good. Yeah. So when you get this problem that you are not getting a very characteristic growth curve, you start blaming the media. The first is the media. Maybe my media is outdated, the media is not okay, my pH is not okay, etc. Make sure that you start with the correct numbers. Sometime what we do is, especially in the microbiology, you streak your plate, right? When you make a pure culture. And then the colony size will differ. Now, if your colony size is too small, and that very small pinpoint colony, you have taken it up and you seeded it in, say, 500 ml culture, you will not see that growth because that initial number or the quorum is too low for it to reach. It will have a very long lag phase. So what you do is get your number correct to get. So you don't blame your media or your antibiotic. Antibiotic is not produced because you are working with the wrong number. And most importantly, because many of you will do your experiments, that is what I, in fact, tell my colleagues in the UG and the PG, handle your microbes carefully. First thing, rule of thumb, do not do plate-to-plate subculture all the time. You must have your microbe frozen very properly in -80 in a freezing medium. Don't incubate your plate for more than 2 days at 37 degrees, thinking that they're immortal. They may grow anytime. No, bacteria ages. We have evidence that bacteria also age at 4 degrees. Aging bacteria is going to lose its property. Sometime if I come there, if I get a chance to come to your university, we will take some series of lectures. Then you will know that why aging bacterial population must be considered. So you have more aging bacteria, yeah? So then you will see that the community is collapsing. So never do plate to plate, never do subculture from tube to tube. Always take a fresh culture, which is 24 to 48 hours incubated culture, and store your cultures in -80. If you have liquid nitrogen, very good for long-term storage, but for short-term storage, store at -80. This is a rule of thumb. Please follow that.
Any question from students?
So, no questions from anybody? Any students? You can always put it in the chat also, but I don't see anything in the chat. Okay, they are not standing. Okay. Sure. Yeah. Thank you, Dr. Goel, for this opportunity, and thank you, colleagues. Feel free to write to me if you have any doubts. Thank you so much. Bye-bye. Take care.